So yesterday when we left our worms, they kind of looked like this. We had transferred them onto a high salt environment. They weren't moving as much. If we actually go back to here, we can even see that a lot of them look kind of shriveled up. And I should have mentioned that this was 15 minutes after we moved them from the low salt environment to the high salt environment. So the point of this video is to understand what was happening to the worms over those 15 minutes after they got moved to a high salt environment. Bringing some understanding to what's going on, we had an environmental stimulus. So this is, if you think back to the gene regulation notes, this is talking about those signals when we say, you know, the body receives signals. So the signal was from the environment in this case, and it was that we transferred the worms from a low salt to a high salt environment. And then because of that stimulus, the worms had a biological response where through osmosis, the worms got seriously dehydrated and right to the point where they weren't even moving. Some of them, they looked really shriveled up. So if you remember, osmosis is the movement of water from low osmotic pressure to high osmotic pressure. So because there was so much salt in the environment, the water moved out through the worm and dehydrated them. And so that's really what was going on with the worms. So now we're going to look at the worms about 48 hours later. What we know is that in the high salt environment we had dead and dying looking worms. So now let's take a look. And as I said, that video was taken about 15 minutes after moving the worms. So now let's take a look 48 hours later. These are the exact same worms that we saw before, and we see they are now a lot happier. There's actually a lot more there. They've clearly done some reproduction in their time off. This is one of them moving through their food. You can kind of see some of their intestinal tract in there. But we see after 48 hours, they're looking pretty spiffy again. That looks like they have no problem with this high salt environment anymore. So let's take a look at what that means. So as sort of a recap to the data, in the high salt environment, again, after about 15 minutes, they looked like they were dead or dying. But then 48 hours later, they seemed to have rebounded. They seemed really happy again. So think about what this could mean for what's going on in the worm, and then there is a glycerol reading online that I want you to read and then answer the questions. Glycerol, if you remember, is one of the components of triglycerides, which we learned about in lipids way back in like October. So we're going to look specifically at that glycerol molecule right now. And so then you're going to do the glycerol reading and answer the questions, and then you can move on to the video that's part 3. But again, maybe you do that tomorrow.